It's my pleasure to introduce our next faculty, Jingyuan Chen. So Jingyuan is faculty here at the Martinos Center. I see several members of our lab here. Welcome. And so Jingyuan doing a lot of like multimodal imaging. You should ask her after the end of the session about her combined EEG PET MRI, also working on a lot of high resolution fMRI at 7 Tesla, an expert in physiological noise and physiological networks in the brain. And so we're really happy to have her here to tell us about physiological noise across cortical layers. Jingyuan, thank you so much.
Yeah. Okay. Yeah. Thanks for the introduction and also inviting me to present in this very interesting laminar from my short course. So following Lucas' talk about thermal noise, now we're going to talk about like physiological noise. But actually for my talk, probably have a more theoretical focus instead of actually how we're going to correct for that. But we will get to that point. So first, just to put everyone on the same on the same page about physiological processes. So we all know that fMRI can track internal or external stimulus-driven hemodynamic changes. That's kind of the major contrast for fMRI. But as a living human beings, there are lots of autonomic processes going on in the body. So now you are breathing, your heartbeat, your heart is beating, and also there are blood pressure changes. So all these kind of systemic physiological processes can also trigger hemodynamic changes at different temporal scales. And also your body and tissue movements coupled to these physiological processes can interact with fMRI signals through different mechanisms. For instance, your lung expansion can cause field perturbations that also affect the fMRI signal magnitude, or the direct motion can also disturb the dynamics of fMRI signals. And in some cases, your autonomic states can also interact with neuronal states. So essentially, these systemic autonomic processes can interact with the local neurogenic fMRI responses through various different mechanisms. Well, in many cases, we call those kind of noise because they're not what our signal interest. But I also want to give you the impression that just from this figure, you can get how these line of this physiological signal, physiological noise is kind of blurry. It really depends on specific study purposes. So given that impression, so for the goal of this lecture, first is I will probably give a brief overview of all those kind of common physiological noise people think that can confound fMRI interpretations. And then answer the question, why do we care about these physio noise for laminar fMRI studies? And towards the very end of this lecture, I think you probably already heard a lot about thermal noise or physio noise, dominant regimes. Which physio noise are people talking in this context? And also in thermal noise dominant regimes, do we really need to care about this physio denoising? So hopefully toward the end of the lecture, we may have a consensus on these topics. So because I'm kind of have a signal processing background, so I tend to view things based on their frequency distributions. So here, just a kind of separation of these physio noise based on their temporal scales. So directly, you can see the respiration peak and cardiac peaks. So these are kind of coupled to this process in a very high frequency range. But there are also very slow processes in the autonomic process. For instance, your heart rate has very slow variability, and your respiration are not constant across time. And these kind of slow variations in systemic physiology really trigger hemodynamic changes in very low frequency scale. And actually, in some literatures, you may hear people referring to those neurogenic fMRI responses, not of your primary interest as physio noise as well. But we won't go into too much detail into that particular category of noise, just to be mindful. So first is like those instantaneous cyclic physio noise. So this is just plot the movie of these raw fMRI intensities. You can already spot those intensity and shape changes of the signals in the images. And these cyclic fluctuations are more obvious if we just focus on those ventricle and sagittal sinus regions shown here. So what are the causes for this instantaneous cyclic physio noise? There are actually multiple mechanisms. So the first is directly dynamic partial voluming, just the tissue and CSF movements related to the cardiac pulsatility effects, which you direct spot from the previous image. Another mechanism is that when there is respiration and cardiac cycles, there will be fresh arterial blood that will alter the concentration of deoxyhemoglobin. So this is a kind of major contrast for fMRI imaging. So you will also see these kind of cyclic fluctuations. So for signals arising from these two mechanisms, they are quite localized to very large vessels. So for instance, this is just a characterization from the human connectome data, and then you can see those regions have the strongest influence from these cyclic fluctuations really localized to those kind of large vessels. So I point to a few regions, for instance, insula and some brainstem regions, these really are close to those vessels, and you need to be careful about this video denoising. There are also other mechanisms, because some people probably can tend to have respiration coupled head movements inside scanner. So that pattern really kind of depends on the specific subject and how it's going to show up in fMRI signals. And the final one is related to your chest expansion coupled to these kind of respiration processes. So for instance, if you look at the face image of the brain, and then you can see it really tracks the cycle of the respiratory traces. And it has a very specific spatial signature. So basically, as the place kind of decay away from the chest, the influence are going to get more minor. So this image you can show here is really the base of the brain has the strongest influence. So we have reviewed these kind of typical types of noise. They're in such a high frequency range. Why do we care about for fMRI? And actually, for laminar fMRI, we have learned from Saskia and Renzo's talk and many others, it's like we always have very long TR acquisitions for these fMRI data acquisition, especially probably for laminar fMRI, quite common to see that. So there's such an aliasing effect. So basically, even your original signal is very fast, but you sample at very long TRs, and this signal are going to alias into low frequencies, kind of interfering with your fMRI signals. So now we move to the second type of physio noise, which is in a much slower range. So as I mentioned, you may have like variations in end-tidal CO2 level or fluctuations in the respiration volume and depth. And because CO2 is a vasodilator, so when you have fluctuations in CO2 levels, they will also cause changes in blood flow pumped to the brain and change in the hemodynamics. And also blood pressure and also heart rate variability will also change the blood pumped to the brain and later affect the fMRI signals. Is it working? Okay, great, thanks. As you may notice, like the mechanisms of these slow physiodynamics really mimic these neurogenic responses. So that actually suggesting their spatial temporal features are very similar to those neurogenic effects. So this is actually probably one of those seminal studies call people's attention to this very slow systemic physiology. You can see that it's really confound lots of gray matter areas. So this is a case where they correlate the brain signals to the blue curve, which is a little bit like the envelope of those respiratory traces. So this kind of reflects the respiration volume per unit time, and then it really accounts for variance in lots of parts of the brain. And this is another early study where they try to correlate heart rate variability with global like fMRI signals, and you can see strong couplings at different temporal lags. So not just spatially, their temporal characteristics are also very similar to the stimulus-driven fMRI responses. So here are just some examples of people try to characterize these kind of impulse physio response functions, a little bit like these impulse canonical hemodynamic response functions. So I wouldn't go into details how they characterize it. So basically, you can notice that their timing, I have the 5-second kind of scale showing the bottom. It's really kind of a little bit similar or approach the shape of the canonical hemodynamic response function. So, but while probably a lot of literature view this as physio noise, I also want to make a small caution here. It's like, while these kind of systemic global fMRI dynamics have different mechanisms or different from those local neurogenic fMRI responses, but they're still regulated by the autonomic nervous system, right? In some cases, so the interpretation or separation can be tricky when your autonomic states track dynamic neuronal states. So I just show an example from actual data from my results. So this is just one example where the top just shows the global fMRI signals you measure during the course of a scan. And you can see there are some places where you have a huge dip in these fluctuations. And then the second line is showing the respiration volume per unit time estimated from the respiratory kind of traces, you can clearly see every time there's huge dip there, there's actually corresponding physiological effects. So in this case, probably anticipate removing them, you will kind of improve this neuronal specificity of your fMRI signals. But here is another subject's data where you can see this global signal seems so tightly track this kind of physiodynamics. In this case, I'm just wondering whether subjects fall asleep inside scanner, there's a global state changes. So I think just a caution that in the specific cases when your physio processes kind of likely track the states or track the ongoing task stimuli, we need to be very cautious about viewing any fluctuations as noise and projected out of the brain. Okay, so we have reviewed all these kind of physio noise. I'm listed on the right of the column here. And actually from Camille's talk tomorrow, you may learn more about this, how this cross-layer vasculature and how we model fMRI signals from that. But basically, I want to take a little bit more interactive so we can go by them one by one and then to identify which of these sources probably we're going to show these layer-specific dependent patterns going to interfere with these layer-dependent neuronal responses. Okay, we go first. The first is like the head motion kind of coupled to this respiration process, the really displacements of the head. So how many of you do you think it will kind of really manifest as a specific layer dependent patterns? You can shaking your head or nodding your head if you get an idea about your votes. Oh, you think it could be? Oh, yeah, I think that probably would be where it depends specific region and the rotational angles. Yeah, probably a little bit harder to direct interpret it to be consistent across layer patterns. Yeah, and the second is like the cardiovascular pulsatility. It's just those big muscles movements. Yeah, people are nodding. It's because like the large PO vessels in the surface of that really have the largest kind of CSF and tissue movements. Yeah, so actually that's probably the one quite relevant for laminar specific features. And how about this kind of deoxyhemoglobin concentration changes coupled to cardiac and respiratory cycles? Yeah, so people are nodding. Yeah, I think that's the case because across layer there are different vessel densities, and then so all these going to modulate the signals. And the last one is like chest expansion, just causing the main field change. Are there specific layer dependent patterns for it? It's a mixture of nodding and shaking. I think that could really, a complicated thing. It kind of really depends on which part of the brain, and because there has a specific spatial signature of the how distant it's from the chest, and then it really depends on the layer geometry and the interaction with that distance. So not that very straightforward. And how about this slower scale bold noise, kind of causing changes deoxy? Yeah, they are nodding. Yes, just basically the same mechanism as those direct cardiac respiration induced changes. So actually by doing here, you can notice like the influence on laminar profiles are not based on the specific mechanism, the kind of frequencies more related to the specific mechanisms kind of driving these changes. So first is like for this dynamic partial voluming, we agree it's kind of strongest in the PO vessels, right? So ideally, you were expected the patterns to decay as function or go to deeper depths. And if you have very small work sources, you may be able to see a kind of elbow pattern or sharply decay as it deviates from these PO vessels, and then a little bit more flat when you go to this white noise. And I believe for many fMRI mechanisms, probably all have these particular concerns. And second is about these deoxyhemoglobin concentration induced mechanisms. So this kind of really depends on the laminar vessel density. And I think John already showed how these vascular layers can vary across different regions and not homogeneous across layers. So this is kind of showing a very nice early study where they contrast these layer-dependent physio profiles to these kind of vascular density maps. You can see a good alignment with that. Certainly how the layer manifests really vary across different regions, but that kind of gave you an idea from the physiological perspective what these these kind of layer-dependent deoxy effects should manifest. Another thing I want to mention, while these kind of mechanisms comprise of both those fast oscillations and very slower scales, those cardiac and respiratory cycles may not be that efficient in driving fMRI responses because they're just in such a high frequency range. So even for this mechanism, the dominating factor is still those ultra slow fluctuations seen in your data. And also this just from a physiological perspective, and you have actual measures, right? And different sequences have different spatial sensitivity. So that kind of actually will drive how you're going to observe these kind of deoxy patterns in your data. So for instance, if you don't do any kind of profile alignment or whatever further processing, and for gradient echo bold signal, it's going to see a more obscured kind of patterns of these layer manifestation, the profiles, and basically a little bit quite like those neurogenic responses you capture with gradient echo bold, and you can see a little decay of the signal go to deeper depths. Okay, so I'm going to show a little bit examples because there's not too much literature in this domain, so probably a lot of them just using gradient echo bold, so you can get an idea about our patterns. So first is like the different patterns can actually be imaged by layer-dependent studies. So for instance, we probably wouldn't go into too much details about the mechanism. So basic idea here is that if a vessel is signals arising primarily from those deoxy concentration mechanisms, then its magnitude are going to change as a function of its orientation relative to the main field. So in this study where it's actually by my previous colleague, Olivia Wiseman, so they look at the orientation dependent profiles of these instant respiratory variation and those cardiac pulsatility. As shown on the left, the different line here, different cortical depths. So we can just pay attention to the blue line, which is kind of above the pure surface. So you can see it really clear shows a kind of orientation dependence. Kind of this is more like a correlate, it kind of suggesting it's kind of really deoxy effects. Where you look at those variants explained by the cardiac pulsatility, it doesn't show this kind of orientation dependence. So while this, as I mentioned, these respiration effects are so small compared to the dynamic pulsatility and other effects, but it can still really read in the real data. And here is just an example of kind of average across like 12 subjects and different brain regions and how these physio noise are explained by those kind of instantaneous cyclic noise. So you can really see how it decays as a function of the cortical depths. And also compared to the profiles kind of explained by much slower scale, if I'm not quite imagining it, it kind of probably suggests a pulsatility dominance because it has much faster decays and also kind of much stronger, like above the pure surface, kind of shown here. Another thing you might notice is like I really show a voxel size dependence. If you contrast those orange lines into green and blue, you can really see these effects kind of get much smaller when you reduce the voxel sizes. So this kind of arises from several mechanisms. I think it's probably covered by all those different lecturers. One is like when you have smaller voxels, you can really avoid those big peel vessels. So the number of voxels getting affected by these dynamic partial volume effects or those large vessel size effects get smaller. And another is just like we're in the wide noise dominated regime, the fractional contribution of physio noise gets smaller, which we will get to a little bit in a few slides. And another thing I feel probably haven't been kind of appreciated enough is like just in terms of the relative ratio of signal to the physiological contrast noise. The idea here is like when you have several voxels adjacent, the neuronal, the physiological signal may tend to be more similar across those voxels compared to these neuronal dynamics. So when you have a larger voxels, it could be these more severe cancellations of those instantaneous neuronal dynamics compared to these very slow scale physio fluctuations. In that case, when you have a larger voxel, you're probably going to decrease the contrast of the signal to the physiological noise. Okay, great. Another thing I also want to highlight, not just like their spatial, the profiles across depths might change a bit. Actually, for different sources, we already highlighted a little bit. It's like they will really affect different spatial distributions. I don't think people usually use like voxels as large as a 1.5 millimeter to really look at depth-dependent profile. I'm here just using that to help you visualize where the regions are affected by these different physio processes are. And then you can see like for cyclic fluctuations, fluctuations is really in the vicinity of those large vessels on the left. And on the right side, for those very large systemic physio, more like posterior of the brain, including lots of sensory regions, that actually has been shown by many studies. So because of these systemic physio effects really have the depth-specific profiles, and also they have the cross-region variability. So we probably would expect to confound those laminar fMRI activation and connectivity. But surprisingly, there haven't been too much studies really directly characterizing this. I probably just used an early study from John to highlight this importance of just suggesting how this might alter these connectivity results. So in this case, I think he probably focusing on a specific patch in the visual cortex and then perform a within column connectivity. So basically dividing into 11 depths and calculate the cross correlations. And then on the left, just showing the raw data without visual signal removal. And on the right side, he showed after preprocessed following this physiological signal removal. And then these results are initially maybe a little bit counterintuitive because you see an increase of these fluctuations. It sounds like these physio noise are masking out those true correlation results. And then the interpretation for this data will likely be these physio effects are just not homogeneous across layers. And then, or maybe less homogeneous compared to those neuronal effects. So that's why projecting out will have an enhancement in the dynamics. Of course, whether this will generalize to more data, different contrast, and more brain regions kind of remain for the test. But at least this it's kind of very powerful proof that these systemic physio or layer-dependent physio profiles may going to affect this kind of neural dynamics a little bit. So to summarize a little bit, is we first kind of review some common physio noise that probably you see a lot in fMRI literature that may confound the interpretations of fMRI signals. And why it would be relevant for layer profiles actually is kind of depending on the mechanisms. So for dynamic partial volume effects, just strongest above the pure surface and decay much faster as you go to deeper depths. And for those deep oxy concentration effects, it can occur at different frequencies, but it's really kind of more dominated by those slow systemic effects. And from physiological perspective, we will scale according to the specific layer vascular profile. But how you show up in the detected image will depend on the acquisitions and also the preprocessing you were doing in your data. And because they have the specific layer profiles, and then if you're not removing them, they likely provide a confound in your laminar fMRI data. And finally, if you want to remove this noise from the data, surely when you go to smaller work sizes, it's going to mitigate these effects a bit. And also you can use model-based or data-driven approaches to address this noise. But we also mentioned at the beginning of saying how it's going to affect this, what the type of physio noise we're talking to when we're talking thermal or physio noise dominated regimes. So I have to say, it's quite vague definitions of physio noise in the context of thermal versus kind of physio noise dominance across literatures. And probably can correct me if I'm wrong. The earliest paper probably proposed this concept is from the Kruger and Glover paper in 2001. So I went to the literature and just see the definition of this physio noise in the context. So basically lump sum of everything, including those neurogenic responses out of interest in these particular cases. So if we have the consensus of what's the definition of this particular physio noise, so do we need to address physio noise in thermal noise dominant regime? I think there's no simple answer to that. It's very clear probably we don't need to care about that. The justification here is the contrast to noise ratio is signal and versus the combination of physio noise plus white noise. And ideally in a case where your white noise dominated and then I think the sign of physio and white noise should be kind of reversed. So the contrast noise ratio can just be approximated by your original raw signal and the white noise. So in this case, probably you think physio noise are not a huge concern for our data. I just want to mention probably two kind of exception or some caution to this kind of case. In one case is probably your data acquisitions in the thermal noise dominant regime, but did you do a lot of crosswalk averaging and in your smoothing in your data preprocessing. In that case, probably you're pushing data back to these kind of physio noise dominated regimes. So this is just one thing you need to be mindful about. And the second is like, all the discussions are primarily focused on the noise aspects of the data. So what about signal? How confident are we going to separate a signal of interest from the potential physiological sources? One thing we need to be mindful that the contrast of signal to physiological noise remains roughly similar, although there may be due to those aggregation effects I kind of mentioned. So briefly, I don't think this will be a huge effect. For instance, you want to see task evolved effects, or you have very clear definition of your signal and use that to regress signals across layers. So probably wouldn't be a huge concern. But imagine a case, for instance, you generate the signal of interest from the data itself, like from ROI or whatever from this data, probably still kind of benefit from the signal or regressing out this physio noise. So it really kind of depends on specific data. And finally, if you want to learn more about this physio noise, here's some more literature on that. And there's a video recording talking about those model-based denoising approach. And toward the very end, I just want to mention one point related to the question asked yesterday about ICA denoising, and briefly check with Renzo before the talk today. I think the case is is whether ICA denoising will be helpful in removing physio noise in the laminar fMRI data. I think the first question is that whether you were thinking denoising is necessary for your specific applications, right? We mentioned the case where you have very clear definition of the signals, and also your data are pretty short in the white noise dominated regime, probably wouldn't be a huge concern. But in other cases where it could be a concern, how are you going to identify the signal? I think also separate into two cases. In one case, is there going to have a large coverage of your brain? Because in that case, probably ICA decomposition is going to be driven by the kind of things in the typical domain of the large kind of vessels or whatever. So you can still extract these kind of different physio profiles, and then you project out the data. So there's nothing special about laminar fMRI. But there's also another case, actually probably credits to Renzo. It's like if you have very big fixed field of view, very smaller, just to specific cortical layers, right? Now we think about the assumption of spatial ICA. It does have a constraint of spatial sparsity. And you imagine the case where the patterns of neurogenic responses and patterns of spatial, these physios have very overlapped spatial patterns. Probably your ICA would not work in resolving them separately. In those cases, probably temporal ICA or other more fancy approaches could be applied, but that's just a potential thought for these ideas, but not really being validated. But it's certainly quite an interesting question to talk. Okay, great. So that's all the talk for this lecture. So I don't know how much time I'm taking Doug's time, but happy to take any questions. If not, I'm happy to answer them at the end of the lecture.